So, hi everyone. So I'm very happy to introduce the second student speaker. By the way, very nice talk, Yogi. Debanik from my group. Debanik, most of you might know because he was the vice president of GSA. But what you don't know is that he's a very talented experimentalist in the lab, doing some very interesting work that he will talk about here today. Debanik came here from the University of Jadavpur in India as a master's student, and he did very well in his master's. He decided to move into his PhD, and I'm very happy that he did so because he did some very good work. He received several awards over the years, including an award from the American Aging Association and from the CBE department here, the Best Poster Award a few years ago. So Debanik's work is mostly in the area of aging, which is a bit off the beaten path for chemical engineers at least. So that's what he will talk here today. Several years ago in my lab we found a transcription factor. It's called a molecule that we can use to reverse aging in cells in culture. So Debanik continued this work that other people, and he got much deeper into it. And today he will tell you how that molecule works and how there may be an opportunity to actually develop through that understanding small molecules that might do similar work to reverse aging at the cellular level, but also at the level of the animal, of the mouse at this point. So without any further, Debanik, let's get Debanik.
Hello everyone. You can hear me right here. So thank you, Professor S.Andreadis, for that kind introduction. So the topic of my presentation today is inhibition of glutaminolysis, that is glutamine breakdown, restores mitochondrial function in senescent stem cells. So as we age, the cells in our body, they go into a state of stable cell cycle arrest. That is, a percentage of cells, they stop growing. And this phenomena is known as cellular senescence. So these senescent cells, they tend to accumulate and secrete factors that affect the neighboring cells and tissues and disbalances tissue homeostasis. Hence, accumulation of the senescent cells is harmful for a healthy aging. But what causes this cell senescence? One of the major hallmarks leading to cellular aging is mitochondrial dysfunction, and that is what my research is focused on. So what is mitochondria? To answer that, let's talk about how a cell works. So we can imagine a cell to be like a village or a town. And what does a village or a town require? It needs manufacturing units. So these organelles or functional units, they make proteins and nucleic acids that are required for a cell to function properly. It needs transportation or infrastructure that helps things to move around in a cell. It requires distribution, and these organelles, they sort out components and they send it out and they move them around to where they need to go, inside the cell or even outside, like international trade. It needs a trash bin, and it needs an IT hub, and nucleus being the IT hub of a cell. It contains DNA, which is sort of a blueprint for everything. But for all these functions to work properly and harmoniously, the lights need to be kept turned on, and for that, that's where mitochondria comes into play, and that's what I'm going to talk about today. So this is how a mitochondria looks like in red, and this is a cell. These are densely packed around the nucleus of a cell, and these are double membrane bound. There's outer membrane and an inner membrane. And along the inner membrane, there are 5 complexes. And electrons gets transported from complex 1 through to complex 4, which is the major oxygen consumer. And 95% of the oxygen in a cell is actually consumed by mitochondria. At the same time, when electrons are getting transported through the complexes, protons are pumped out through complex 1, 3, and 4. And this creates a voltage gradient along the inner membrane called MMP, or mitochondrial membrane potential, and that enables the ATP synthase to form ATP. So what happens when this mitochondria fails? So the importance of mitochondria in the context of aging was demonstrated in this elegant study where the authors, they generated a mitochondrial depleter mouse model. They deleted the mitochondrial complexes and they found severe hair loss, wrinkled skin, and high age of mortality, both in male and female mice. But their most interesting finding was when mitochondria was repleted back in the same mouse, it grew back hair and became healthier. And this study thus shows that the decline in mitochondrial function promotes aging phenotype, and if we can restore mitochondrial function, aspects of aging can be reversed. So in our lab, to study aging, we isolate mesenchymal stromal cells from human hair follicle, and we keep them in culture. We use a cultural senescence model where we keep the cells in culture for about 50 to 60 days till they lose their growth potential. That is, after this time, the cells stop growing, they become big and flat. And we term these cells as senescent or aged cells. And this is how the cell looks like. These are young cells, much smaller in size, and these are the aged cells, much bigger in size. Now what we do is, we express a gene called NANOG in these senescent cells. NANOG is an embryonic transcription factor, and what I mean by that is it's expressed in the embryo, but in adult tissues, NANOG is not expressed. That is, we don't express NANOG. So we express this gene in the senescent cells via a DOX regulatable system, where addition of this chemical, doxycycline, enables NANOG expression within 24 hours. The aged cells expressing NANOG, we term them as SN. The young cells, we term them as for Y, for young, and senescent cells as S or aged cells. Previous work in our lab, just a few studies. My former colleague, Panos, he showed that the intermediate filaments that are important for transportation or for maintaining the cell structure, actin and ACTA2, are highly downregulated in senescent cells. And when you express NANOG, it is restored back. And this was done in 2D system. Later on, another former colleague of mine, RF, he showed in 3D culture, when he seeded the cells in a micro device, the same observation that held true even in 3D environment, where NANOG restores actin and ACTA2, intermediate filaments important for transportation and cell structure. And also the contractile force is restored by NANOG expression. A couple of years later, Na showed some of the hallmarks of aging are reversed by NANOG expression. So there are several hallmarks of aging, and some of them are like DNA damage, which can be visualized by this stain in the nucleus, gamma H2AX, that was immensely decreased by NANOG expression. Also, cell cycle genes like P53 and P21 were downregulated by NANOG expression. When I started working on this, I found another hallmark of aging, that is ROS accumulation, which is a hallmark of metabolic dysregulation that was also suppressed by NANOG significantly. And this data actually prompted us to delve deeper into what NANOG is doing and how NANOG alters the metabolic landscape of the cells. So the first thing, we looked into the mitochondria of the cells. The MMP, or mitochondrial membrane potential, was restored by NANOG expression, shown over here. Also, the mitochondrial subunit COX-4 protein expression also went up with NANOG expression. But this is just a protein expression. To check the functionality of mitochondria, we employed an instrument called Seahorse Analyzer, which we have in our lab. And how this works is by sequential addition of drugs that inhibits the various mitochondrial complexes, it gives out a respiratory profile like this, where the output is oxygen consumption rate. And from this profile, we can measure ATP-linked respiration, which is equivalent to ATP production in mitochondria, and spare capacity. This is a very important measurement, and spare capacity, as the name suggests, is a spare energy, which a cell does not use in its basal state, but if there is a sudden demand, then they can use it. And this is an indicator of mitochondrial health of a cell. So we compared the young, aged, and the NANOG expressing cells, and we found that the senescent or aged cells severely diminished ATP-linked respiration, and NANOG increases that. Also, spare capacity was also increased with NANOG expression. Thus, we can conclude that NANOG in fact rejuvenates the mitochondria of these 8 cells. Now we know that mitochondria is restored. We looked into other metabolic pathways like glycolysis, which happens in the cytoplasm of the cells. And we observed that the GLUT receptors, which takes in glucose, goes up with NANOG expression. Also, glucose uptake significantly goes up. Also, CHOS analyzer, from that we can also get glycolysis, and that is also upregulated. So we know that NANOG is restoring this pathway as well as this pathway. What about the coupling of this glycolysis to mitochondria? Glycolysis can be coupled to mitochondria via this enzyme. This is called the gatekeeper enzyme, pyruvate dehydrogenase. When pyruvate dehydrogenase is phosphorylated, it uncouples the two pathways. We found that NANOG dephosphorylates the pyruvate dehydrogenase, thus coupling glycolysis and mitochondria together. So this shows that NANOG alters the metabolic landscape significantly, but expressing NANOG can be a challenging issue as it is a genetic manipulation. So we wanted to see if from NANOG we can figure out what pathways are affected, and we can target those pathways instead of expressing NANOG. So we looked into our RNA-seq data, and we looked into all the transporters that are affected in our RNA-seq with NANOG expression, and we found the most upregulated transporter was SLC7A8. And this SLC7A8 is related to glutamine efflux. Other transporters like SLC38A2 and SLC7A7 are related to glutamine influx. Those are downregulated by NANOG. So our RNA-seq hints at the fact that NANOG might be affecting the glutamine pathway. So to confirm that, we looked into the enzyme that breaks down glutamine, and that is glutaminase. And we found significant upregulation in the 8 cells in the RNA level, and the enzyme activity was high. Also, the protein level was very high for the enzyme that breaks down glutaminase in the senescent cells, thus showing that these 8 cells are heavily reliant on this amino acid glutamine. And NANOG suppresses that. Now, since these cells are breaking down, or the 8 cells are breaking down so much glutamine, which is an amino acid, we looked into the byproduct of amino acid breakdown, and that is urea. And we observed significant upregulation in the 8 cells, urea accumulation. Intracellular urea was significantly upregulated. Now, when we inhibited the enzyme by a small molecular inhibitor, CBE839, we observed that the urea level went down in senescent cells almost to the level of the young cells, thus showing that most of this urea is coming from glutamine breakdown. So our next question was, can this intracellular urea accumulation impair mitochondrial function? To prove that, we knocked down urea transporter. This transporter is on the plasma membrane, and it works by means of facilitated diffusion. So it takes out urea from a region of higher concentration to lower concentration. So I knocked this down, this transporter, in young cells, and I forced the young cells to accumulate urea, and then I checked the mitochondrial function. And yes, the mitochondrial function in fact goes down. The membrane potential went down, ATP-linked respiration and mitochondrial health went down significantly when urea was accumulated. On a side note, it was interesting when we looked into the RNA-seq data again, we found that urea transporter was actually the most downregulated transporter in 8 cells. So it's not just that the 8 cells are breaking down amino acids, it's also that they are accumulating amino acids because they cannot get rid of the urea. So far, we see the senescent or 8 cells, they have high glutamine breakdown. That leads to high urea accumulation, and we found that urea accumulation can lead to decreased mitochondrial function. But this is a correlation. To establish causality, our next hypothesis was, what if we inhibit glutamine breakdown? Can that improve mitochondrial function in the 8 cells? For that purpose, we employ 2 modes of intervention. One, we treated the 8 cells with the inhibitor CBE839 for 7 days, and we also knocked down the GLS enzyme. And we found in both modes of intervention, urea accumulation went down. And that was associated with increased mitochondrial membrane potential and increased ATP-linked respiration, and the spare capacity and index of mitochondrial health was restored back almost to the level of the young cells with the inhibition of glutamine. Thus, we can conclude over here that inhibiting glutamine pathway can restore mitochondrial function in the 8 cells. And this restoration of mitochondrial function was associated with decreased aging hallmarks like DNA damage, ROS or reactive oxygen species accumulation, inflammatory cytokines production like interleukin 1 alpha and beta. So, most of the mechanistic insights so far that I presented was in MSCs. We now wanted to see if our findings can be extended to other aging models or disease models. So we looked into progeria or EGPS, Hutchison-Gilford progeria syndrome. These patients, unfortunately, they live for around 14 to 16 years, and they have an accelerated aging phenotype. So what happens here is there's a mutation in this gene, lamin A, and that compromises the nuclear architecture and promotes aging. So we got fibroblasts from these patients from Progeria Research Foundation, and for control, we used fibroblasts from the father of the patient that did not have the lamin A mutation. And we observed mitochondrial function going down in the progeria fibroblast, and this is in line with other reports in literature as well that have reported the same thing. But what we found was this was associated with increased glutaminase activity and increased urea accumulation. For our in vivo system, we used the similar progeria mice called lackey mice, and this progeric mice, they live for around 14 to 16 months instead of normal 30 months. And we did our experiments when the mice, they were 10 to 12 months old. And we isolated heart and skin tissues, and we found COX-4, which is in red, mitochondrial COX-4, was significantly down in the skin tissue and as well as in the heart tissue. Again, this was in line with other reports in literature that has reported the same thing. But we observed that this decreased COX-4 and mitochondrial expression was associated with increased glutaminase activity and increased urea accumulation, both in the skin as well as in the heart. So our next idea was, can we replicate our findings in vitro to in vivo model? That is, if we inhibit glutamine breakdown in vivo, can we see mitochondrial function getting restored? For that purpose, we used progeria mice, both male and female, and we injected this mice with the CB839 GLS1 inhibitor thrice a week for a period of 1 month. And after 1 month, we did some behavioral analysis, and a week later, we sacrificed the mice and we isolated mitochondria from heart and skin tissues. And again, we used the seahorse analyzer, and we observed complex 4, the mitochondrial complex 4 activity was significantly improved with glutaminase inhibition, both in the skin as well as in the heart, and that was associated with decreased ROS accumulation in skin and heart tissue. Finally, we also did some behavioral analysis to check if the overall health of the mice is improved, and for this purpose, we used some geriatric assessment tools. This is gait speed, which is the speed a mouse takes to walk from one point to another in a straight line. And we found that the treatment group, the gait speed was significantly improved. To check the endurance of the mouse, we hung the mouse upside down on a grid, and we recorded the time it takes to fall down. And we set the time limit as 5 minutes according to our IACUC protocol, because going more than 5 minutes can stress the mouse. And we found that the treatment groups, almost all of them, they did not let go of the grid for 5 minutes. So hanging endurance was also significantly improved with this inhibition or with this drug administration. Another very simple yet very effective way to check the overall health of the mouse is how well a mouse can build a nest. So for that purpose, we gave raw materials for them to build nest at night when the mouse is most active because they're nocturnal. And after 12 hours, we went back and we checked how well they build the nest. And then we scored the nest, and we found that this lackey mice, without the treatment, they were not able to build very good nest. They made like cup-shaped mostly, while the treatment group, their nesting ability improved quite a lot. And thus, all this shows that inhibiting glutamine breakdown can improve, not only restore mitochondrial function, it can improve the overall health of the mice as well. So finally, in conclusion, we observe severe mitochondrial dysfunction in the AH cells. AH cells, they have increased reliance on glutamine, the amino acid. Increased glutamine breakdown leads to urea accumulation, and that impairs mitochondrial function. Now, NANOG suppresses glutamine catabolism and restores mitochondrial function. So we use NANOG as a tool over here to come up with another small molecular inhibitor, CBE839, that blocks this pathway. And we observed by using this enzyme, using this inhibitor, mitochondrial function is restored in the AH cells. And our in vitro findings held true in vivo system as well, where administration of this drug increased the mitochondrial function and overall health of the mice. And an interesting fact is CBE839 is actually an anti-cancer drug. It's actually an FDA-approved trial 1, if I'm not wrong, human trials. And here we showed that this anti-cancer drug can also be used in the context of aging to reverse mitochondrial dysfunction associated with aging. And that's about it. The final slide, which is actually the most important slide, because I have to acknowledge people. Dr. S.Andreadis, obviously my professor, my mentor, who has been always supportive of my work. And the best thing is, whenever you have a doubt, you knock on his door and you'll get an answer. Each and every one of my lab mates, almost half of my lab mates are co-authors in this work. So it was a very collaborative work that we did, and I'm grateful to everyone. Also our collaborators from Jacobs School of Medicine, Ram, Ken, Dr. Tron, especially Ram, who taught me a lot about metabolism and how to run seahorse analysis. And finally, my lab partner and my best friend, Na, who graduated last year, now a senior research investigator at Beijing Research and Development Center. Equal credit goes to her as well for this work. And of course, the funding sources, because we need money. And thank you.
Yes. So this is very exciting work. This drug that you talked about, would the potential plan be for you would need to continue dosing it forever in order to maintain that mitochondrial function, or do you have any idea about whether it might be helping to restore more physiologically and you wouldn't need to maintain those doses, or there might be some other long-term effects?
Yes, that is a very good question. So we did not remove the drug. So ideally, to answer your question, we should have treated with the drug and removed the drug to see if the effect continued. We did not do that, so I'm not sure about that. Yes.
Yes, please. How about with the nano? Did you remove dots?
Sorry, what? Can you repeat?
With the nano expressing cells, when you remove the dots, it would stop producing.
Yes. Yeah.
Was the effects lasting, or was it?
Yes, I did not do that work, but RF did, and he removed NANOG, and the effect lasted for at least 8 days. But eventually it went down, slowly. Yes. Yes, please.
Nice talk. Can you speculate why senescent cells would rewire to go for glutamine crisis?
Yes, that's good. So we see in the AH cells, glycolysis is not working properly. We also see mitochondrial function severely impaired, but they must need another source to maintain their bioenergetics. It can be that by rewiring the glutamine pathway, because ATP can also be produced in the TCA cycle. And it has been shown in other literature that when there's a lot of mitochondrial dysfunction, there's succinyl-CoA synthase, the enzyme, that makes a lot of ATP to compensate for the loss in other pathways. So I'm just speculating here because I don't have the data, but it's possible. Yes.
So you mentioned in one of your earlier slides that mitochondrial dysfunction, along with mitochondrial dysfunction, there are other factors that contribute to overall aging. So could you speculate as to, like, let's say you just solve completely problems of mitochondrial dysfunction, how would that benefit overall aging? Like how?
Yes, that's also a very good question. So each of us, like the research labs, they usually focus on one thing. It's not only mitochondrial function that is affected. There are other pathways that are affected as well, and they are all interconnected together as well. So just by improving mitochondrial function, I'm showing that over here, some aging hallmarks are reversed, but overall aging may not be reversed. The other pathways also need to be, included. So yeah.
Let's think.